Let us have the third presentation by Dr. Sandra Habicht from the Institute of Nutritional Sciences, Justus, Liebig University, Giessen, Germany. She is a postgraduate researcher with 9 long years of experience with Gourd research. Now over to Sandra.
Thank you, Prima. I'm a nutritionist, very much interested in nutritional physiology, so I'm trying to really make this understandable to the audience and to those people who do not have a medical or nutritional background. This is about the anti-diabetic effects of the bitter gourd. It's a lot about biochemistry and the mechanisms behind the anti-diabetic effects of the bitter gourd. And it's not my own results, it's more or less a literature review. We had a chance to publish a review paper in 2013, and this is part of this. To start a short introduction, I would like to say a few things about bitter gourd and diabetes mellitus. Bitter gourd, we have talked about this already, it's an important market vegetable, and it is a medicinal plant that is mainly used for blood glucose control in different populations. And this is a study conducted in Malaysia. It's not a study, it's a survey. And in Malaysia, bitter gourd was named as the #1 plant that is used for diabetes as complementary medicine. And bitter gourd is in the internet very present. When you type diabetes mellitus plus bitter gourd, you will have lots of results in Google, for example, more than in PubMed. So there's also a lot of non-scientific literature available on bitter gourd and diabetes, because there are a lot of beliefs and myths around bitter gourd and diabetes. And there are products that are sold in the internet and also in the markets. That are promoted as anti-diabetic, and they are made from bitter gourd leaves or bitter gourd fruits. There are teas, juices or capsules, tablets available in the internet. You can buy them for more or less money, but usually the products, they are sold without any information on safety, on how much you should use for what kind of blood sugar level, and there's very little information, because the information is also not really known by the scientific audience. Scientific information on bitter gourd we will find, for example, on PubMed. And in PubMed there are around 170 results when you type bitter gourd and today. There's also scientific data available on bitter gourd in cancer research for hypertension and dyslipidemia and other disorders, but the scientific focus is on the anti-diabetic effects of the bitter gourd. And in the literature, the scientists, they agree upon the fact that the bitter gourd is very high in nutrients and also very high in bioactive compounds. So this is just to show how the scientific interest in bitter gourd and diabetes is increasing. In the 60s, there were only a few publications, 1, 2, and so on. And now, recently, the interest has increased, and there are very many publications per year in the last years. So this is a table to show you bitter gourd compounds with anti-diabetic effects. We have heard a lot about bitter gourd compounds with anti-diabetic effects already, and when you read the papers, they all mention compounds found in bitter gourd that are related to diabetes and anti-diabetic effects, but there are actually only very few studies available that really prove anti-diabetic effects for defined compounds. So that means there are only very few data available where they really isolated compounds from bitter gourd, and the isolated compounds were really tested in cell culture or animal models to prove the anti-diabetic effects. And this is only these few compounds that I found. This is the P-insulin or V-insulin, that stands for plant insulin or vegetable insulin. Later they named it polypeptide P. This is a protein which is very similar to the bovine insulin, which has only one amino acid different from the bovine insulin. And this has insulin-like effects also in humans. These are triterpenes, which have proven anti-diabetic effects. There are more isolated compounds, fatty acids, and other compounds that have really proven anti-diabetic effects. And I don't want to go into detail for the compounds, but as you can see here, there are already some mechanisms mentioned what these compounds are doing. For example, we see here there's an increased secretion from the intestine of GLP-1, and this is the glucagon-like peptide 1, and when this is released from the small intestine, it will increase the insulin secretion into the blood and will help blood glucose control after meals. And there is also one other thing mentioned here, for example, the PPAR alpha and gamma activation by beta-Gourd compounds. PPAR alpha and gamma are very much involved in fatty acid and glucose metabolism in the body, so very important results here. So as you can see, there are few different mechanisms on how beta-Gourd is helping to lower blood glucose levels in diabetic patients and subjects. And these are results from cell culture and animal studies that I have summarized here. There are three major pathways on how beta-Gourd can reduce blood glucose levels. This is the intestinal glucose uptake can be reduced by eating beta-Gourd or drinking beta-Gourd juices. And there are two major findings published in animal studies. This can be like inhibition of the starch digestion, or the glucose uptake can be reduced itself. And there can be an increased insulin secretion, for example by the GLP-1 secretion of the small intestine, and there can be, this should be a separate point, the pancreatic beta cells can be protected by beta-Gourd antioxidants, so the pancreas is able to secrete more insulin. And insulin sensitivity of muscle cells and adipose tissue and also the liver can be increased by reducing overweight in individuals. PPAR gamma activation, the insulin signal can be improved, PTP1B can be inhibited, and insulin-like peptide is present in the beta-Gourd, so this will also help take up glucose in the peripheral tissues, and there can be an AMP kinase activation, which will have a glucose uptake of the cells, which is independent of insulin. So I will have a few examples from the literature for each of these points. I will start with the glucose uptake by the gut. There's one slide missing, so I will go to the second example. This was published in 2007. This group, they used healthy 8-week-old rats. They were fasted overnight, and then they were given beta-Gourd extracts orally, the water fraction, or extracts from the water fraction, and in addition they were given 1 gram of sucrose per kilogram body weight or 1 gram of glucose per kilogram body weight. And there was for each, for glucose and for sucrose, there was also a control which did not receive beta-Gourd extracts but just distilled water instead. So what we could see from the results is that when the rats, they took up the glucose load from, or the sucrose load from the oral administration, of course, a few minutes afterwards, the blood glucose level will increase after glucose feeding, and the beta-Gourd inhibited only the glucose concentration increase after sucrose feeding. That means when you gave to the healthy rats glucose in addition with beta-Gourd, beta-Gourd did not have any effect. But beta-Gourd together with sucrose inhibited the glucose uptake of the rats from the sucrose. That means the sucrose was not digested, and beta-Gourd inhibited sucrose digestion. In other studies where they used diabetic rats or mice, They could also lower the glucose uptake from glucose administration. But this is only when the mice are really diabetic, because then the other mechanisms will come into consideration. I will have examples for the increased insulin secretion after bitter gourd administration. This is the first example published in 2005. They used an animal model. This is type 1 diabetic rats, and they got water extracts of bitter gourd seeds, and they received the treatment 30 days at a dosage of 150 milligram per kilogram body weight. And the results were that the bitter gourd firstly prevented the weight loss. In type 1 diabetes, usually the subjects will suffer from uncontrolled weight loss because the glucose levels will increase, and then lots of the glucose will be excreted by the urine, and also they don't have the insulin, which is an anabolic hormone. So the bitter gourd prevented the weight loss in diabetic individuals. Here you can see this is the control group and their weight gain during the experiment. The type 1 diabetic mice without bitter gourd treatment, they lost weight, and the bitter gourd treated type 1 diabetic rats, they kept their body weight stable. In addition, they measured blood glucose and HbA1c levels, which were improved significantly. Also, what they did was an oral glucose tolerance test, which was also improved significantly. And there was also, they took the liver tissue, and they found there is a lower activity of enzymes in the liver that will prevent the body from storing glycogen. So there was also higher glycogen concentration in the liver, but which might not be a bitter gourd effect. It can be an effect of an increased insulin secretion. The reasons for the increased insulin secretion after bitter gourd treatment might be, for example, this group wanted to look into this, might be a protection of the insulin-producing cells in the pancreas. In this study from 1998, they used male type 1 diabetic rats that were treated with bitter gourd juice for 9 weeks. And afterwards, they took the pancreas tissue and analyzed the tissue, and they could show that the insulin-producing cells, they were very low. This is the black spots in the pancreas. This is the insulin-producing cells. There were only very few insulin-producing cells in the diabetic control. In the non-diabetic control, the insulin was present in the pancreas. And after bitter gourd treatment, the type 1 diabetic mice had significantly higher number of insulin-producing cells in the pancreas. So that they assume that bitter gourd with antioxidants can help protect the pancreas from loss of insulin-producing cells. The third one is from our colleague, Professor Wang, from Taiwan, and this is on GLP-1 secretion from the small intestine. This is a study they conducted in cell culture study. They used small intestine cells that can secrete GLP-1, and when incubated with bitter gourd, the GLP-1 secretion was significantly higher. They also tested the fractions, the water fraction and fraction of small molecules from the water fraction in mice, I think, and could show that the bitter gourd increased GLP-1 levels in the blood, and at the same time the insulin and decreased the blood sugar levels. Last but not least, there's the improved glucose tolerance from peripheral tissues. That means from, as I mentioned, muscle cells, adipose tissue, and the liver. And this can be improved. The glucose uptake of these tissues can be improved by different pathways. Professor Krawinkel this morning was already talking about the inhibition of PTP1B, which can be one reason for an improved insulin sensitivity, because PTP1B, the protein tyrosine phosphatase, is somehow stopping the insulin signal at the cell level. So when PTP1B is inhibited, insulin can work better. And we found that in type 2 diabetes, the expression of and activity of this enzyme is increased due to overweight. PPAR gamma activation will also have an improved effect on insulin sensitivity. There are also oral drugs available that increase PPAR gamma activation. Beta-Gourd is very good because PPAR gamma activation can go ahead with increased body weight. But Beta-Gourd is also at the same time activating PPAR alpha, which will control the body weight, so they will not have the negative side effects of weight gain. As I mentioned, the insulin-like peptide, which can be the plant insulin that can be extracted from Beta-Gourd, can also improve glucose uptake from peripheral tissues. And I have one example published in 2008 for a better glucose uptake in peripheral tissues. This is a study with female type 2 diabetic mice. To induce type 2 diabetes, the mice were fed with a high-fat diet, which was like 58% of the calories in the diet were from fat, and they had 1.5% of a Beta-Gourd juice added to the diet, and the mice were fed this diet for 16 weeks. And after the 16 weeks, the body weight was lowered by Beta-Gourd compared to the control, and there was a normalized plasma glucose compared to the control, which did not receive the Beta-Gourd. The authors also in this study, they did an insulin tolerance test and also a glucose tolerance test. A glucose tolerance test is where you give glucose orally and measure the glucose increase in the blood for a certain time, and the glucose tolerance is improved when the blood glucose doesn't rise so much. And insulin tolerance test, you will inject insulin and see how the mouse responds to the insulin with decreasing of blood glucose levels. And in this study, there was an improved glucose and insulin tolerance, and at the same time there was an enhanced insulin response, which they found in the tissue. This is the liver tissue. They did what they did was they were looking at the signal at the insulin receptor, because the insulin in the blood will bind on the insulin receptor of the liver, and then there will be a signal, and the signal can be measured, and this is what they did. This is a control which is not insulin resistant. They will have a high signal after insulin is bound to the receptor. The high-fat diet control, they will have a lower signal, and then after Beta-Gourd treatment, the mice had almost normal insulin responsiveness. Conclusions from in vitro and in vivo studies are we learn a lot from the excellent studies published in the literature from cell culture and from animal studies about the anti-diabetic mechanism of the Beta-Gourd. However, the results are usually highly significant, and the study designs are made for highly significant results. That also means that authors and publishers, they tend to really increase the dosages of the Beta-Gourd in animal studies to something that is not able for humans to consume. And of course this is important because we learn a lot about the mechanism and if Beta-Gourd is effective or not. However, it's still difficult from the animal and cell culture studies to really have a dietary recommendation, and which is also not known is what is the most effective plant part, dosage or preparation of the Beta-Gourd, because there's hardly any study where these things are compared in different treatment groups. However, what we can assume from the literature published is that the anti-diabetic effect of the beta-gourd results from a complex action of multiple compounds. That means, for me, for example, that the whole fruit might be better than just the flesh, just the seeds, or just an extract. So really to benefit from all the compounds that are present in bitter gourd, we should consume the whole fruit as it is and not discard any of them. There is proven effectiveness in cell culture studies and animal studies, but what about humans? Is there also the same evidence? I feel there is in some studies, in others not. And this is an overview of human studies. I have 2 slides, so this is only the first part of it. There are different designs. We have placebo-controlled, we have not controlled studies, we have very few participants, we have more than 100. Is the 142 I think is the maximum participant number in the human studies with bitter gourd. Study designs, they differ very much, and also the outcome measures. The studies are hardly to compare because also the bitter gourd that is given is different in every study. They use commercial products, they use extracts, they use juices or just tablets. I would like to present this study. This was done with 42 patients in Taiwan. This study is very interesting. It was not only looking at diabetes, it was looking at the metabolic syndrome. The incidence rate of the metabolic syndrome could be reduced by bitter gourd treatment with 4.8 gram bitter gourd per day in tablet form. The whole food powder was given, but they did not have significant results on blood glucose levels. However, what they also showed is they measured complications. They measured indicators in the blood that would show and indicate toxicity of bitter gourd, and they could prove that this dosage, which is relatively high, is safe for humans. That is why this study is so interesting. And there's another study with 60 type 2 diabetic patients I would like to talk about, because sometimes the question comes up, is it better to consume raw or cooked bitter gourd? And this is the only study who compared raw and cooked bitter gourd products. They had a mixed powder of bitter gourd fruit with fenugreek seeds and yambu seeds. They mixed the powders, and then they had a capsule prepared from this, which represented the raw bitter gourd, and they had a biscuit prepared from this powder, which was a baked bitter gourd powder, a baked plant powder, which should represent the cooked bitter gourd. And they could show that the raw bitter gourd was more effective. This is just to also mention the cooked and raw difference in human studies. This is the only one I found analyzing this one. So with this, I would like to thank you for your attention and ready to take questions.